Some people ask themselves why I sometimes write case reports. I just like it because, you know, we are individuals and medicine sometimes is individually too. So here is a little case report. Actually, Doug Richman, a case study. The aim of this study was to identify factors for success of Doug Richman, a highly successful infectious disease physician scientist. The methods working with him intensely 1996 to 1999, and thereafter I conducted an observational study of n equals 1 over 20 years. All right, now the results. He's an incredibly bright person, thinks multidimensional, identifies new research questions rapidly, bridges clinical to basic science, attracts great people of all sorts, except of me, for example, to form multidisciplinary teams. He lets people thrive and develop their own ideas, lives scientific freedom. He's a great writer. He corrected lots of manuscripts of myself, but they would look totally red, of course. And he attracts a lot of funding. He's incredibly efficient, takes care of his people, not just with regard to research, and he remains modest despite success. So in conclusion, he's an outstanding, inspiring mentor, an idol for me and many young and old researchers, many of them among us. Thank you, Doug. So I think the paper by Joe, Chelsea, Marjan, Carol, Doc, myself, and others changed the life of many people in science. Of course, as often in science, we were not the only one. There was another one at the same time by the Silicono group and by John. But I don't talk about the quality. Ours was the best, of course. Now, this data, you know, the decay of the viral outgrow data from the Silicono group, showing that the half-life of the reservoir measured by this method is about 44 months, so it's a long time. We will not eradicate HIV with antiretroviral treatment. Another important paper, I think actually a very important one, was the one by Matt, Joe, and others that showed that if you treat early, actually you do something to the reservoir, and that's still the single thing we can do today, early treatment. And actually we showed this also when we looked at some cellular expression of unspliced RNA, and many other groups looked at this and has been replicated many times. Now the first part of the data I present you has been published recently. It's a large study basically based on the Swiss HIV cohort study and also a little bit on the Zurich primary HIV infection study. That's kind of an acute cohort that I started in Zurich 2002, and this one is much older. I just give you a little impression what the Swiss HIV cohort study is. Actually, I'm the president of this undertaking, and it's probably my only presidentship I will have in my life. So it's organized by having 7 centers, 5 university hospitals, 2 large cantonal hospitals. Then we have a lot of private physicians and smaller hospitals affiliated. We have typically Swiss decentralized biobank. And then we have interactions, we have a data center, and we have a coordination center. And I think that the nice thing about this cohort, it's running since 30 years. It has more than 20,000 patients enrolled, and actually we know something about those patients. You know, it's not just some patients. And we actively follow 10,000 right now, and we have all transmission groups, so there's no selection going on. Everybody who's HIV infected and above 18 can enter. We have 25% women, and we have 75% of all HIV-infected people on antiretroviral treatment in Switzerland enrolled in the study. And so far, there were more than 1,100 publications, and of course, many collaborations and stuff. But it's just something that is still thriving. And so what is a cohort? It's actually basically a collection of individual data. You have many patients, you start to follow them at some point, and then you are collecting data in a systematic way. We collect clinical data, demographic data, lab data, genetic data, behavioral data, drugs. We look, for example, are people smoking? What co-medication do they have? What's risk behavior do they have? Do they have non-HIV related diseases, co-infections, and so on and so on. And of course, you adapt this to new questions and you get new data. We have also a lot of adherence data, we have resistance data, and so on. Now, this study was basically the foundation of the work we did here. So we wanted to identify the determinants associated with HIV reservoir size and long-term dynamics in the Swiss HIV cohort study. So we included patients who were on antiretroviral treatment and never failed for at least 5 years, meaning always below 50 copies. I come to the blips and the low-level replication later. No treatment interruptions, no virological failure, and of course we needed samples from the biobank. At least 3 at 1.5 years, 3 years, and basically 5 years. We didn't care about the first phase decay because that has been studied a lot, and we were not interested in non-integrated DNA, and Joe colleagues have shown that in the first year, basically all non-integrated DNA decays. So we measured proviral DNA, not very sophisticated, but I think in 1,000 patients you cannot do sophisticated stuff. And so we had 1,057 patients, and then we looked at, because we had a lot of patients, I mean this is 10 times larger than any decay study ever done in HIV, we could look at different factors, covariates, treatment, gender, ethnicity, age, time on ART, time of untreated HIV infection, time to viral suppression, CD4 cell count, pre-treatment, transmission group, subtype, plasma HIV RNA, and blips, or low-level replication. Here is the definition. A blip was allowed once above 50 copies, or low-level viremia once above 200 and then below 50 and 200, and that was a failure twice above 200 copies. Now here is the study flow chart. I'm not going into detail, it just tells you it was pretty systematic. You need a lot of patients to come up with, at the end, with 1,057. Some baseline data, I'm not going into all. We had about 20% non-white people. We had 25% females, and we had about 50% men having sex with men, and 1 to 3 years of non-treatment we had in 27% of the cases, and 5 to 7 years untreated, 26.5. And you have to realize this is historic data. Today we treat everybody. You will not get this data anymore. So this is, again, something in a cohort that you can go back in time and look whether these factors have had an impact. Then we also compared the treatments. We just had NNRTI and booster PIs because integrase inhibitors, of course, were not available to have a long follow-up at that time. And we had about 70% subtype B, but still about 30% non-Bs. So here is the first set of data. We see here Beanplots, 1.5 years, 3.5, 5 years, and 10 years, because we had 412 patients who had a follow-up of 10 years. And as you see, there is some decay, but of course a huge variation. But each time point following the other was significant, so there was a clear decay. And actually, to me, a very striking finding was that not that overall there was a decay but of about a half-life of 5.6 years, but that in 26% there was actually an increase in DNA. And these are patients treated as good as we can. And I didn't, first I thought it was wrong, but it was not. And the slope 1.5 to 2 years, of course, is smaller, is longer, 11.8 years. I mean, these half-lives are pretty much in the ballpark we know. And again, here 24% had an increase, so even over 10 years. And of course, one can speculate what's the reason. I come back later to this. And actually, we did quite some tests whether this is true, not just some assay variation. It's really it's there. So we saw a kind of a plateauing effect after 5 years. So after 5 years, not much is going on. And now I think the question was, do we find determinants influencing the decay, the reservoir size first, and then later on the decay of the reservoir. So here we just look at the 1.5 year. Is it higher or lower? That's the question. And we looked at all these covariates I showed you in the beginning, and I just show you the ones we saw an effect. Actually, viral blips and low-level viremia were associated with higher reservoirs, so there was clear effect, meaning if you had in this time period before 1.5 years some blips or low-level viremia, you had a high reservoir. And as you know, clinicians often don't care about blips, you know. I mean, it has been shown that blips are actually associated with virological failure in large studies, so we know that, but they even influence the reservoir. I don't know how, but we can discuss that later. Then we have seen that the pre-treatment HIV RNA is associated with higher reservoir and time to viral suppression. If it's just wait forever, reservoir is higher. And then also a negative effect, if you have a higher CD4 count, you have a lower reservoir, and initiation of antiretroviral treatment during acute, within 1 year, was associated with a low reservoir, and non-white ethnicity actually was also associated with lower reservoirs. And people having had an infection by using drugs, as injection drug use also, but this is a survivor bias, I'll show you later. So overall, I think the data makes sense because several of these parameters were known, and I think that's true. We also looked at some Fiebig stage cases because a reviewer asked us to do that, and we saw that the early ones, Fiebig 2, were the lowest, and then some Fiebig 4, Fiebig 6, they were just still clearly lower than the average. So again, it makes sense. Low reservoir if you really treat early. We know that, but still we see it in the population, and that gives us some confidence that this data is good. Now the question is what's happening with the decay. Now we look at this period. So basically we did slopes. We generated slopes of these time points, whether there was faster decay and less rapid decay. And what we found again was the blips and the low-level viremia, they were associated with a slower decay. And the CD4 cell count pre-treatment was associated as the higher with a faster decay, so the immune system helps. And the non-B subtypes were associated, at least in a multivariable model, with faster decay. Time to viral suppression, time on antiretroviral treatment didn't play a role here. So when we looked at low-level viremia and viral blips, we found that they were about twofold less steep decline as inducing. So I mean they had about twofold lowering effect in this model. Now, the relationship between these blips, I mean, the blip thing was really kind of the major thing, I think, in this study that came out, that they are of some virological relevance. And it's quite a complex interaction because blips are associated with replenishing the reservoir and slow down its decay. But the size of the reservoir predicts blips thereafter. So it's quite a complex thing. It's clear. I mean, this is clear, but the causality, of course, we cannot infer from this data. And so there are different ways to explain it. One could be its immune activation with clonal expansion that basically where cells actually also produce virus, or another scenario that there is low-level replication and some rare events of infection of some cells, and that we see this virus production. Then we also looked a little bit more into the subtype thing, and we found that, as I told you, that non-B subtypes have a low reservoir after 1.5 years. Here, just individual subtypes. Of some, we have very few. And we actually did an analysis just including white ethnicity, because we had 581 with subtype B and 133 whites infected with non-subtype B, so we could kind of exclude the ethnicity thing. And we still saw this effect, so it seems to be something there which is affecting the size of the reservoir, meaning that the Bs are higher. I just want to show you the enrolled patients, because I told you that the people living, also having had intravenous drug use as a risk factor or transmission group, had a survivor bias. And here you see in the early days there was quite a lot of intravenous drug users enrolled, but that stopped about here in the late 90s, because the harm reduction programs really kicked in, and of course the ones who survived from here, they had a good immune system and were slow progressors, and that's the reason they have lower reservoirs. Now we also looked at drugs. NNRTIs boosted PIs, there was no difference. And I just show you a graph I really like. I always like to look at raw data. And what you see here is the history of treatment of 1,057 patients. Here, boosted PIs, you see they start here, and then they change to other drugs, often to NNRTIs or other boosted PIs if the green color changes. And below the NNRTIs, this is wrong, and you see that they change a little bit less. And but overall, all these patients were always below 50 or had a blip, or as I showed you. And just tells us you can sit quite a while looking at these graphs, because there's a lot of individual data in. Anyway, so there are some strengths in this study. It's highly representative cohort. We have a lot of cofactors that we can study and correct for. And it's a large study that looked at these factors. Of course, it's observational data, and you cannot really make causal inference. Potential for unmeasured confounding is always there, and we cannot differentiate between replication competent and incompetent viruses. So in conclusion, the slopes of total HIV DNA are negative on average, but positive in 26.6%. Which I still find remarkable. The decay slows down over time and approaches a plateau after 5 to 6 years. Blips, early treatment initiation, non-white ethnicity, time to viral suppression, and transmission group of infecting drug use, intravenous drug use were associated with low total HIV DNA values, 1.5 years after ART initiation, and blips, low-level viremia, were associated with high total HIV DNA values and also strongly predicted the decay of the viral reservoir. So we found this complex interplay, and this needs further work to resolve. But I must say I find it relatively difficult to just explain this by clonal expansion, I have to say. Because if you think about it, you need quite some population of cells, you know, clonally expanding and then producing virus in a kind of a synchronized way, which I find not so easy to explain. And probably it's a mix of things, as often in medicine, the black and white doesn't often not exist. Anyway, now we come to the last part, and that's much shorter. It's heritability of the HIV reservoir size and decay under long-term suppressive treatment. I mean, there have been many studies done to look at different phenotypes, looking at kind of the contribution of the viral genome, explaining the variation in those phenotypes. And actually, we did a lot in the cohort of those studies. So we looked, and others, at the viral set point, and over the years, because there were many studies, about 20 to 30% of the variation of the viral set point is explained by viral genome. The CD4 decline, it's about 10 to 20%, and the antibody imprinting capacity, meaning whether a virus, a similar virus, induces a similar antibody response, being either BNAbs or binding antibodies, was estimated to be 10 to 15% in recent work by us. And as a comparison, host genetics explain approximately 15% of the variation of the viral set point. So overall, I think the virus has some impact. And now we wanted to see whether this virus impact, also the virus genetic impact, can also be seen in decay and the size of the reservoir. So it was just kind of an additional variable that we were studying. So again, we looked at 1.5 years and then at the decay. And how can you do that? Actually, it's not, I find it quite complex. The idea is not, it's kind of not complicated, but how you do it, it's quite some statistical exercise, which of course this is not really my field or expertise, and I ask me every day what my expertise is, and I don't know. The older I get, I think I don't have any expertise, so that's great if you have some people who know what they are doing. Anyway, so what we did, we used the mixed effect model and other models too later, but I don't go into those. But what you basically do is to construct a phylogenetic tree, and actually I learned the first ones from Andy Lee Brown. And then we had partial pol sequences in 869 patients and whole genome sequences in 475. So we generated those trees, and what you basically do, you look at clusters and you look whether they have different phenotypes, meaning a different, let's say, different decay if you have similar viruses, and you can play around with the distance between those clusters. And then you feed this data in the model. So we feed as fixed effects, transmission group, sex, ethnicity, basically all the factors we had. And then as a random effect, you had these transmission clusters. And what we also looked at was at the evolutionary models. And I'm just because it's quite complex, you need different models. And I'm not going into those now. But what we found first, that when we looked at the reservoir size, so basically here at 1.5 years, that there was an effect of heritability. Here we played around with the distance threshold, so if you got more conservative, actually the heritability became larger. And also looking at the other models I didn't explain, there was an effect, not a big one, but there was one. So overall, I think we are confident to say that there is a heritability effect on the size of the reservoir 1.5 years after. When we looked at the near full NGS sequences, before that was partial pol, we still see an effect. Just at the very stringent distance threshold, we don't see it, but we have very few clusters here. But again, it's consistent, so I think there is something there. Now, when we looked at the decay, there were some mixed signals, so this is not as clear as the other one, and of course, we don't have the perfect power to look at it. Besides, I mean, despite the fact that we have a lot of patients, we still don't, maybe not have enough, but anyway, there is some signals there that it could be, so we would say it's kind of a trend. So overall, we found a consistent and moderate heritability of HIV reservoir size 1.5 years after therapy initiation, and a more tentative heritability of the HIV reservoir decay. And if the reviewers are as nice as people in San Diego, we might hopefully also get a positive review in some weeks. We will see. Anyway, I showed you some recent work, and I thought I have to talk about reservoir things here because, yeah, for me at least, it started here, definitely. And many people were involved. Of course, the patients participating in the study, in the cohort, nurses, doctors. I mean, these things are really amazing because you need big networks, motivated people. You don't make money out of it. I'm often surprised that it actually works. But it's only because people are interested and they realize it's good for the patients because we learn a lot of things. And it's also quality control, of course, how we treat patients. And the major people here were actually Nadine Bachmann, a PhD student, and Chenjie Wang, a master student who did basically all the analysis, and Roger, who is I would say, our statistical bioinformatics guru, and Cory Metzner, who developed the proviral DNA assay, and of course all the funders. Thanks. And maybe at the end, it's really, I think this is exactly the type of work I learned in San Diego, you know, to have different people involved and yeah, and just be part of a team, and actually that was taught by Doc to me.